In this video, we're going to introduce the general procedure for Salmonella isolation. The purpose of this lab is to detect the Salmonella from foods. Two methods will be used: rapid detection method and conventional method. Rapid detection method is an immunoassay which will be discussed in details in the next session. The conventional method could be separated into 4 steps: pre-enrichment, selective enrichment, isolation, and identification. Remember, food matrices sometimes could be very stressful to bacteria. For example, low pH of juice and limited iron access in egg white. Food processing could also result in injured cells. For instance, heat treatment, high-pressure process. Those injured bacteria will not survive well in harsh selective media. That is why we add a pre-enrichment step before selective enrichment step. At pre-enrichment step, a non-selective medium will be used to allow the recovery of those injured cells so that they're healthy enough to endure a subsequent selective enrichment step. The non-selective medium also allows the growth of the bacteria so that the population is increased to detectable levels. However, the medium used in pre-enrichment step is usually not nutritionally rich, so that it does not cause the overgrowth of background microbiota. Next step is the selective enrichment. The selective broth media used in this step allows the growth of Salmonella but suppresses the competing microbiota. 2 kinds of selective broth will be used to increase the chance of recovery, because some strains might survive better in one broth, not the other. The purpose of isolation is to obtain single colonies on selective and differential agar plates. Whether the presumptive single colonies obtained from isolation step is the targeted pathogen will be confirmed through identifications. Identification steps could include serological tests such as anti-Salmonella antigen, molecular-based tests such as 16S rRNA tests, or biochemical tests such as sugar fermentation. Those 4 steps are not only used in Salmonella detection but also commonly used as a guidance to design the detection protocol for other pathogens.